Alright, so we're going to start up with our first lecture here on meiosis, which is still involved in the same lecture slides that we had when we did the mitosis unit, but now we're on to meiosis. So this is actually chapter 11 in the Raven book, or the book from the bookstore. So, with this particular section of chapter 11, the very first thing we need to do, the first thing we need to look at is why meiosis needs to happen during the sexual life cycle. So to think about that, what we have to first think about is what fertilization is. Right? So fertilization is the combination of two cells from two individuals that join together to form a zygote that eventually becomes a full-fledged, well, first an embryo and then eventually the full-fledged organism. So if you imagine taking a diploid cell from a human father and a diploid cell from a human mother and join those cells together, you'd have 46 chromosomes plus 46 chromosomes equals 92 chromosomes. So actually in the second generation of this particular species, our own species, or humans, we'd actually have double the number of chromosomes in that next generation. We go from 46 to 92. And then if we had a third generation of humans, so now we're looking at 92 chromosomes plus 92 chromosomes is going to give us 184 chromosomes in the next generation of humans. So before too long, as you can see, the number of chromosomes in the species is going to get out of hand if we double it every single time that there's going to be a new generation. So what we want to do before we have fertilization is actually take the cells from the parents, cut the chromosome number in half, and then allow them to redouble during fertilization. So if we take dad's cells, 46 chromosomes each, cut those chromosome numbers in half to 23, do the same thing with mom, so now we have 23 chromosomes plus 23 chromosomes gives us 46 chromosomes, which is the correct number for our species. So meiosis and fertilization, all we're doing there literally is cutting the chromosome number in half and then redoubling it to create the next generation. And so if we think about any even number, 20, cut it in half, redouble it, you're back to where you started. And that's exactly what meiosis and fertilization are going to do. So the diploid cells are going to be turned into haploid cells during meiosis. Right? So again, those terms are going to come back and visit us again. Diploid cells are those that have two sets of chromosomes. Haploid cells are those that have only a single set of chromosomes. And so because we're animals, our cells are going to have two sets of chromosomes, one set from each parent, 23 chromosomes per set, for a grand total of 46 chromosomes per cell. When we create our gametes, which is what we're dealing with primarily in this chapter, gametes are our haploid cells. Gametes, again, are sex cells, sperm cells, egg cells. Those are all haploid cells in our life cycle. But in animals, that's the less, dramatic part of the life cycle is the haploid part. It's always single-celled, whereas the diploid part of our life cycle is the rest of our body, which is a large multicellular organism. Haploid cells are always single-celled in animals. So the benefit then of going through all this trouble of meiosis and then fertilization is genetic diversity, is what it boils down to. So we get genetic diversity during meiosis, as we're going to see here in the upcoming slides. We also get genetic diversity because we're combining chromosomes from two separate parents who are unrelated, hopefully. That's going to also create genetic diversity. We also have the opportunity for mutations to take place during this process, which again gives us even more genetic diversity. So that really is the underlying theme here, is that sexual reproduction gives us genetic diversity, which is worth talking about just for a second. Why is that such a big deal? Why do we bother caring about that? Well, by having all of the offspring somewhat different from each other, if you imagine some kind of a new challenge comes along in the environment, maybe there's a drought, or maybe there's a new predator, maybe there's a new pathogen that shows up. If all the offspring are just a little bit different from each other, or a lot different from each other, you've got a better shot of at least one or two of them surviving this new environmental challenge. If all the offspring are exactly the same as each other, then it's a much riskier proposition because your entire batch of offspring might be wiped out by that new environmental challenge. So the diversity is sort of a way for parents to hedge their bets and create offspring that are a little bit different from each other, and therefore hopefully cope with any new environmental challenges that might come along. So this is the basic idea. Again, we have a haploid sperm cell and a haploid egg cell, and those two cells join together to form a diploid zygote. The zygote then goes through mitosis. It becomes an embryo and a fetus and eventually an infant and a child and an adult, and then we go back and restart the life cycle again. So that is what we do as animals. We go back and forth between haploid and diploid, as do other kinds of organisms like fungi and protists and plants. The thing about animals that's different, though, is that our haploid stage is very, very small. Like I said before, it's always single-celled. And the diploid stage is always the more dramatic, larger, more obvious part of the life cycle, as it is in our bodies. So, like I said, the other groups, fungi and protists, they're going to have a longer haploid stage. Plants are more or less 50-50. But animals, which is kind of our focus for this chapter, is by far going to more heavily emphasize the diploid part of the life cycle as opposed to the haploid part of the life cycle. With animals, diploid is the more predominant stage. It's the part that's bigger, made of more cells, more obvious, conspicuous, those kinds of things. That is the diploid part of the life cycle. In animals, the haploid part is the part that's single-celled. So we've seen this picture before. We talked about it back in the earlier part of this chapter where we talked about the animal life cycle as a whole, where we've got adult organisms that are sexually mature producing gametes through meiosis. So egg cells and sperm cells are produced by meiosis. Those get together through fertilization to create a fertilized egg or a zygote, which then becomes an embryo and a fetus and an infant and eventually gets us back to our original starting point, which for a little bit more accuracy, just kind of Photoshop that in there. Okay, weird. Okay, yeah. All right, so now on to meiosis. So the job of meiosis is really two things. It is to cut the chromosome number in half when we're creating gametes, and as kind of like a side benefit, we also get the genetic diversity that I talked about before during meiosis that helps make all the offspring just a bit different from each other. What meiosis is, it is sort of core, is one round of DNA replication followed by 2 rounds of cell division. All right, so if we go back and think about mitosis, what did that deal with? That was, in that case, mitosis entailed having 1 round of DNA replication followed by 1 round of nuclear division. So 1 replication, 1 division gets you back to the original starting number of chromosomes you began with. Whereas meiosis, we're going to do DNA replication once and then divide the cell in half twice. So really, essentially what we're doing here, if you want to think about it in a more just straight up numerical kind of a way, if you take any even number you can think of, you double it, divide it by 4, that's going to get you right back to where you started by half, right? So if we start off with a cell that has 46 chromosomes, if we double that to 92 and then cut that number in half twice, that takes us down to 23 chromosomes. Okay, so that's the idea of meiosis, is double the chromosome number once and then cut the chromosome number in half twice to get us down to half the number that we started with. So 1 round of replication, 2 rounds of division. Meiosis 1 and meiosis 2 are what we call those 2 rounds of division, and there's going to be a couple points along the way, especially in meiosis 1, where we're going to see some features that actually help generate some genetic diversity. Versus mitosis, which is where, back when we covered that part of the chapter, mitosis didn't create any genetic diversity. With mitosis, every cell that you create is exactly the same as the parent cell and exactly the same as any other daughter cells that were produced. So mitosis, the goal, is to generate identical copies of cells. With meiosis, that is not the case. With meiosis, we're trying to create cells that have half the number of chromosomes and are diverse for lots of other reasons as well. So these 2 rounds of meiosis, each has a prophase, metaphase, anaphase, telophase, just like mitosis did. And since we have 2 rounds of meiosis, we kind of call those prophase 1, metaphase 1, anaphase 1, telophase 1, and then prophase 2, metaphase 2, anaphase 2, and telophase 2. There's no round of DNA replication in between them. So like I said before, with meiosis, we're doubling the chromosome number once and dividing it in half twice. Now, something else that's going to be important that we're going to see is a situation called synapsis, which actually happens really, really early on in this process of meiosis during prophase 1, where the homologous chromosomes, the chromosomes you got from your parents, are going to pair up, and they're actually going to become physically attached to each other briefly, and then they're going to do something else which helps to create some genetic diversity. So the synapsis thing is something we're going to see that involves the homologs that you inherit from your parents when you are producing your own gametes. All right, so we'll take a look at that as well. This is basically what we're talking about here. So with these 2 pairs of sister chromatids, maybe this is chromosome number 1. So we have 1 copy of dad's chromosome number 1 and a second copy of dad's chromosome 1. And then 1 copy of mom's chromosome 1 and a second copy of mom's chromosome 1. So what's happening in this picture is that these pairs of sister chromatids are lined up with their homologs. All right, so chromosome 1s from dad are paired up with chromosome 1s from mom. That is synapsis. That actually creates, very temporarily, a situation where those homologs are physically attached together, and as a result of that, they can actually exchange a bit of genetic information, as we're going to see. So that's what we call crossing over. Crossing over is when those homologs actually exchange little bits of genetic information to create chromosomes that are new and unique and novel, chromosomes that have never been seen before in the offspring of two genetically unrelated parents. All right, so crossing over is one of those places where we start to see a lot of genetic diversity showing up in meiosis, and it happens really, really early on during meiosis 1. So again, the idea with crossing over is that during synapsis, the homologs will exchange bits of genetic information, thereby creating almost like a hybrid chromosome, which is different from both of the two parents' chromosomes that we started with. So we'll take a look at that and see how that works as well here. So this is just a really quick example of, first off, a species that's much simpler than our own species as far as chromosome number. This particular species only has 4 chromosomes, right? So we've got maternal chromosome 1 and maternal chromosome 2. Maternal always means from mom, right? And then we have a paternal chromosome number 1 and a paternal chromosome number 2 from dad. So paternal means father's chromosomes, right? So we have chromosome 1 from mom, chromosome 2 from mom, chromosome 1 from dad, chromosome 2 from dad in this starting cell for a grand total of 4 chromosomes total. Before anything else happens, we have to copy DNA. So we do DNA replication just like we talked about back in chapter 14. And so now, in this cell that's ready to go through meiosis, we now have 2 copies of mom's chromosome 1, 2 copies of dad's chromosome 1, 2 copies of mom's chromosome 2, 2 copies of dad's chromosome 2 in that resulting cell. Now we can take that resulting cell and run it through meiosis. So after meiosis 1, the big goal of meiosis 1, and this is one of those things that you should definitely write down, put stars around it, and highlight it and do whatever else makes you remember that it's kind of important, is that the goal of meiosis 1 is to separate the homologous chromosomes from each other. The goal of meiosis 2 is to separate the sister chromatids from each other. All right, so make sure you get that. That's a really helpful way to remember kind of the overall big picture for meiosis 1 and meiosis 2. Meiosis 1 is trying to separate the homologs. Meiosis 2 is trying to separate the sister chromatids from each other. So if we look at this starting cell, we've got homologs together and homologs together. After meiosis 1, these homologs are now separate from each other in different cells. And these homologs are also separate from each other in different cells. So meiosis 1 has separated the homologous chromosomes from each other for both of the 2 chromosomes in these cells. The goal of meiosis 2 then is to separate the chromatids from each other. So during meiosis 2, this long red one goes over here, and this long red one with a little bit of blue on it goes over here. And this short red one goes over here, and this short red one with a little bit of blue goes over here. And then in this cell, long blue goes over here, long blue with a little red goes over here, short blue goes here, and short blue with a little red goes here. So meiosis 2, we separated the sister chromatids versus meiosis 1 where we separated the homologs from each other. All right, so with asexual reproduction, and this is a really good kind of valuable thing to do, is to think about the pros and cons of both kinds of reproductive strategies. With asexual reproduction, you get to pass on, first off, 100% of your genes, which is much better than passing on 50% of your genes, all else being equal. Assuming your offspring have the same chance of surviving, natural selection should favor a strategy where you pass on 100% of your genes as opposed to only passing on 50% of your genes. Another big benefit of asexual reproduction is that you don't have to find a mate. Or if you do have to find a mate, maybe everybody in the population is both male and female, something like that. So that's, again, it's not really asexual reproduction, but it's a bit different than what we normally think of with most animals. But that actually does occur in some animals as well. The big downside of asexual reproduction is that all the offspring are exactly the same. There's no genetic diversity. So if the environment happens to change, and those offspring aren't well suited to the environment, then you might have lost out on a huge chunk of your potential reproduction as a parent, thereby reducing your fitness, thereby causing that particular set of genes to become less common in the upcoming generations. Another benefit of asexual reproduction too is cheaper, right? So you don't have to worry about attracting a mate, finding a mate, keeping a mate, defending a mate. Those are all sort of non-issues if you reproduce by yourself, which is kind of a big one when it comes to asexual reproduction as well. With sexual reproduction, all the downsides are there that were advantages with asexual. So you only pass on 50% of your genes, you have to find a mate, defend a mate, attract a mate, etc. It's expensive energetically, and you're only passing on half of your genes, but the big advantage is genetic diversity. And that can be a huge advantage, which is why most of the so-called higher animals actually do partake in some sort of sexual reproduction. Mutations can still occur in both asexual and sexual organisms, but as we've seen before in previous chapters, they're relatively uncommon, and when they do occur, they tend to be fixed, and even those that are not fixed tend to be either neutral or harmful. So if you're a species that just relies on mutation to achieve genetic diversity, then that's not a very effective way to become diverse as a species. So you're better off, if you need diversity, to go with some form of sexual reproduction. Mutations do add new alleles to populations, but mutation by itself, without anything else to sort of help it along like natural selection, mutation by itself really isn't a very good way to see much in the way of evolution occurring. But some of the things that occur in meiosis do help to mix up the genes, thereby creating novel, what we call genotypes. So a genotype is just the entire set of genes that an organism carries, kind of like a genome. Novel genotypes are produced during the process of meiosis and fertilization that create genetically distinct offspring that might have a better chance of surviving in some sort of environmental change. All right, so something else that we're going to see as we go along. Another factor that helps to create some genetic diversity in species that reproduce sexually is the idea of independent assortment. All right, so what we have on the bottom of this slide are 4 different scenarios. So we've got one scenario here, and then a second one, and a third one, and then a fourth one. What these represent are a species that has actually, in this case, 6 chromosomes. So you can think about having, okay, well, there's 2 long ones, 2 medium ones, 2 short ones. So 2 plus 2 plus 2 is 6. We've got chromosome 1 from dad, chromosome 1 from mom, chromosome 2 from dad, chromosome 2 from mom, chromosome 3 from dad, chromosome 3 from mom. That's a grand total of 6 chromosomes. When we get ready to do meiosis 1, specifically metaphase 1, all right, so you remember what we said the goal of meiosis 1 was, and that was to separate the homologous chromosomes from each other. It turns out the way that we line up the homologous chromosomes on the metaphase plate is entirely random. So in this first scenario, it just so happens that we've lined up all of dads on the top of the metaphase plate and all of moms on the bottom of the metaphase plate. We could also have the opposite. We could have all of moms on top and all of dads on the bottom. We could have these 2 from dad on top, this one from mom on top, and the opposite on the bottom, or any other possible combination of chromosomes lined up on the metaphase plate during metaphase 1. As long as we have a chromosome 1 on top and bottom, and a chromosome 2 on top and bottom, and a chromosome 3 on top and bottom, it doesn't really matter what color they are, because the gamete is still going to get all the genes that it needs. So this is one way that we see a lot of diversity show up during sexual reproduction, during metaphase 1, is with this random lining up of homologs on the metaphase plate. So the second bullet point here, if you think about this, this is fairly striking, right? If you imagine that you could take 2 to the nth power, and that will tell you the number of possible genetic combinations you can have successfully in a gamete to have that gamete be viable. 2 to the nth power, and n is equal to the number of chromosomes in a set of chromosomes for that species. So if we're looking at humans here, without any sort of factor to increase diversity, we actually have 2 to the 23rd power number of ways we can line up our chromosomes on the metaphase plate and still produce viable gametes. 2 to the 23rd power. That's a lot of combinations. And so even without that, we still have crossing over, which greatly adds even more diversity to the offspring that are produced. So what this all kind of boils down to is that with independent assortment and with crossing over, it is actually fairly likely that one individual may never produce the same gamete twice in their entire lifetime. And if 2 parents produce offspring, maybe, I'll give you 10 shots to make offspring, 10 kids, right? That's a lot of kids. But even if you have 10 kids, you're not going to have the same genetic combination twice in the offspring, because there are so many ways that these chromosomes can be recombined and paired up and changed along the way, so that by the time all is said and done, everybody, unless they happen to be an identical twin of some sort, everybody in the population is absolutely genetically unique. And that goes back as far as you want in human history. I guarantee that unless you're an identical sibling of some sort, you have a set of chromosomes that is exclusive to you, and nobody else in human history has ever had it, and very likely nobody else ever will have it in the future of humanity. So this is a huge, huge driver of genetic diversity, is this random lining up of chromosomes on the metaphase plate. It does take a little bit of time to kind of wrap your mind around this, as far as what's actually going on with the homologs and why they're lining up this way. But of course, you can always ask questions. You can always ask me to go back, and I'll cover this again for you. We can show it different ways, but it's just a matter of having the need for just a chromosome 1 on each side of the metaphase plate, doesn't matter what color, a chromosome 2 on either side of the metaphase plate, doesn't matter what color. The color, and the colors, of course, represent which parent they came from. The colors you get don't really matter. As long as you have a number 1, a number 2, and a number 3 on top, and the same thing on the bottom, then it's a viable combination. It doesn't really matter which parent they came from. All right, so getting into fertilization now. If you were to take 2 humans at random from the population, there are approximately 64 trillion different diploid combinations that those 2 parents could produce. Now, to kind of put that in perspective, we only have, and I'm saying only, I'm like air quoting this even though you can't see me, there's only around 7.5 billion people on the planet. That's a lot of people, but still, 7.5 billion is nowhere near 64 trillion people. So you see what I'm getting at there with this huge amount of genetic diversity in the population. You really never see the same combination of genes twice because there are so many different ways of creating unique combinations of genotypes in the population. And by the way, the 64 trillion thing, that doesn't even include crossing over. So by the time you factor that in, this number goes absolutely through the roof with the number of combinations you can have for genotypes produced by any 2 unrelated parents. So again, the ultimate take-home point here is that every single one of us is absolutely genetically unique. You very likely have the only set of chromosomes for our species that's ever existed. The exact combination that you have, the genes you carry, the chromosomes you carry, is very likely absolutely unique in human history. So the next thing to think about before we go into the actual specifics of this is where meiosis happens in the body. And it turns out meiosis only happens in one of 2 places, either in the testes if you're male or in the ovaries if you're female. Now, the overall basic idea of meiosis in both sexes is fairly similar, but there's some really important differences in the number of gametes that are produced, when they mature, which we really aren't going to have a lot of time to go into in Principles 1, but in Principles 2 we do talk about this a little bit more. We talked about the crossing over thing. So right, metaphase 1, the parent homologs move to the metaphase plate, and they orient themselves randomly on either side of the metaphase plate. So as long as we have one member of each pair on either side of the metaphase plate, then it's a valid combination that will work as in a gamete. Since the homologs are moving together toward opposite sides of the metaphase plate, this also means that they're going to move together during anaphase 1 as well. And we'll show some pictures of that to kind of illustrate what exactly I mean by that, because just sort of picturing it is maybe a little bit difficult at this point. So we'll take a look at some visuals here that will help with that as well. All right. So for our purposes, really the more important part of meiosis is meiosis 1. Meiosis 2 is really, in almost every possible way, exactly the same thing as mitosis. So you could even do this. This is one of those points in your notes where, add stars to it, highlight and so forth, circle it. Meiosis 2 is the same as mitosis. They're really indistinguishable. Meiosis 1 is a part that's unique, and that's the part we're going to spend a bit more time because we haven't really seen anything quite like it yet. When we get to meiosis 2, I'm going to put the whole process on the slide at once because it really is just exactly the same thing that we talked about back when I covered mitosis. So meiosis 1, the goal of meiosis 1 is to cut the chromosome number in half. So remember, we're separating homologs, we're separating sets of chromosomes, therefore the cells that come out of meiosis 1 are haploid, whereas the cells that went into meiosis 1 were diploid, the cells that come out of it are haploid. There is no DNA replication between meiosis 1 and meiosis 2. If there was, that would completely defeat the purpose of this entire thing. We do not want there to be any replication between meiosis because that would take us back to our starting point number of chromosomes, which, of course, what we're trying to do here is cut the number of chromosomes in half. So all the second round of meiosis does is to separate those sister chromatids. And if you think back to what mitosis accomplished, it separated sister chromatids. So it looks almost indistinguishable from mitosis. Not I guess technically, but it is very, very similar to mitosis. So that's kind of the point where we're gonna go through this slide, then we're gonna wrap up, and then with the next section that I start on, we're gonna talk about how the steps of meiosis actually work. The cells that go through meiosis are in some ways similar to other cells in the body. They have a cell cycle. They have a G1 where growth happens. They have an S phase where they copy their DNA. They have a G2 phase where they get ready for the actual meiosis to occur. And then they go into meiosis 1 at some point in time when they're directed to by some kind of outside signal. At this point, we have what are called germline cells. Germline cells are those that eventually become gametes, either sperm cells or egg cells. Whenever you see the term germline, that means like a precursor to a sperm cell or an egg cell. And then, like I said before, meiosis 1, there are prophase, metaphase, anaphase, and telophase parts to meiosis 1, and also the same thing for meiosis 2. So not a lot of difference there between what we saw between meiosis and mitosis, at least not yet. So like I said, we're going to stop here with this section, and I'll pick up in just a moment on the next section on meiosis.